Okay, ladies and gentlemen, today we're going to be talking about enzymes. And if you look at our little diagrams here, we know that we're looking at an enzyme. How do we know we're looking at an enzyme? Well, let's look at this one. It's got long chains of proteins all crumpled up and folded on each other. It's got one, two, three, four of them, so we know this is a very complex 3D model of an enzyme or a complex protein. Oftentimes in diagrams, however, our enzymes are going to be represented by these big purple blobs or blobs that have another blob attached to them, but you can see they have certain shapes that match up and pair, indicating that it's an enzyme. So let's go ahead and look at our objectives so we know what we're going to be learning in this video. You're going to be able to, at the end, be able to explain the functions of enzymes in living things, draw and interpret graphs illustrating a chemical reaction with and without enzymes, describe activation energy and how it's affected by enzymes, describe the structure of enzymes, including the active site, substrate, and products. Could you identify those in a picture, in other words? Explain how enzymes bind to a substrate and cause a chemical change. Explain the role of pH and temperatures on enzymes and the other properties of enzymes. Identify the role of inhibitors on enzymes, and in particular, competitive and non-competitive inhibitors. And then how do we name enzymes? So let's begin. Enzymes. First and foremost, they are proteins that act as biological catalysts. What is a catalyst, you say? Well, it is something that's going to increase the rate of a chemical reaction, and it is also going to lower the activation energy needed for a reaction to occur. Well, let's just review chemical reactions real quick before so we can really understand what does increasing the rate of reaction and lowering the activation energy mean. Well, on our last video, we looked at two diagrams like this and was expressing an interest in understanding the chemical reactions that are occurring in these diagrams. Here in this picture, I have reactants that have a higher amount of energy than my products. Therefore, I know energy is going to be released. If energy is being releasing or exiting from the equation, I know it's an exergonic reaction. And therefore, I know I am going to be releasing energy. And it's commonly the reaction in digestion or hydrolysis. In this one, I have my reactants. I have less energy than my products, so I know I'm going to add energy in. So if energy is entering into the equation, it is an endergonic reaction. And we can represent these by dehydration synthesis. So if you look again, we have a molecule that's all bound together. We add some water, we want to separate it out. So we're releasing energy in the process when we break these bonds. That's what we do when we're eating. Remember, we're getting all our energy from our food. We take that energy then and we're going to use it to combine smaller monomers to make larger molecules. If we look at them in a different representation here, we're looking at them on another type of graph. We have my reactants, again, that have more energy than my products. So again, I know this is an exergonic reaction. And over here, I have my reactants that have less energy than my products, so I'm going to add in energy for this to occur. So this is an endergonic reaction. But we're also going to look at these and say, oh, look, they both have a situation where they have to get over a hump. This over the hump is indicating that for any reaction to occur, energy has to be added in some form. And that's not, if it wasn't that way, you'd think that things would spontaneously happen, like water or something could be broken up using automatically, like sugar could break down, salt could break down. But they don't, and that's because they don't have enough energy in them to spontaneously break the molecules that they're made up of apart. So you have to add energy in to get them to separate, or you have to take away to get a change to occur. So when we talk about that adding in the energy for something to occur, we put a name on that. And that name is going to be activation energy. So on this chart, you see here, this bump is the activation energy. And on this chart, it is right here, the activation energy. Both endergonic and exergonic reactions require some energy to get them going. And that would be the activation energy. So when we use enzymes, we say enzymes are going to help lower this little bump to down below. And then in that case, if you're using less energy, you're also speeding up the reaction to occur. So enzymes help reactions to occur by lowering the activation energy, in other words, the amount of energy needed to get them going, and then they help speed the whole process up. So again, enzymes are proteins that speed up chemical reactions by lowering the activation energy. Sometimes you're going to see it in your textbook referred to as E sub A. And by definition, that activation energy is energy needed to start the reaction. So if we look at this graph, we have here, oops, I'm sorry, a line of, the red line is the energy and the time in which a reaction would occur without an enzyme. So it has all the way up to here, it has to climb to have a certain amount of energy, and then it will be able to produce its products. If I add enzymes into this equation, I only need to go up to this height, this amount of energy. So I've lowered considerably the amount of energy necessary to get this reaction to get going and take place. Therefore, I'm also speeding up the reaction. So this is when we talk about how do you draw a chart describing how enzymes work and how they lower and speed up a reaction, is that you can clearly see the red line is the amount of energy needed without an enzyme. This green line down here, and we're talking about potential energy that's going to be required, is a lot less, and therefore it's going to take a lot less time for the enzyme to work. What does that mean in real life time? So if I have a bunch of amino acids and I want to bring them together, you know, I'm going to use an enzyme for them to create all those bonds, and therefore I'm going to create a nice polypeptide. If I wanted to, and that would be considered a dehydration synthesis, which is an endergonic reaction. If I want to take that protein chain then and break them apart, I add in my enzyme, I separate them all out, I have a hydrolysis, and it's an exergonic reaction. Another one we could look at is I have a bunch of monosaccharides. Along comes my enzyme. I'm going to produce a polysaccharide, which again we know is dehydration synthesis. It's an endergonic reaction. I want to take those long polysaccharides then and rearrange them. I add in my enzyme. I separate them all out, which we know is hydrolysis, and an endergonic reaction. Now I could create, you know, this type of animation for lipids and nucleic acids, but I think you get the idea. So let's look at the parts of enzymes then. So if we look at this diagram, here's the enzyme, and you can know and understand that this is the enzyme because you can really see it's not really changing much in the whole entire equation because enzymes do not change. Whereas in this part, I've started out with one solid piece. If I read the equation from left to right, and it's being broken apart here to two separate pieces. If I read the other way, this is two separate pieces becoming one. Again, the only thing that doesn't change in this particular picture is the purple blob, pretty much. And so I know that is going to be the enzyme. The thing that is changing, however, is called the substrate. And that is going to stick to a certain location on the enzyme called the active site. And so when that happens, the substrate is going to bind with the enzyme and create different products. So again, if I read from left to right, this substrate binds with this active site. It's going to create breakage of a bondage, and then I end up with two separate products. If I read from this way, I've got two, this could also be substrates. They're going to come in, they're going to be bound together, and I'm going to produce one product. So the substrate is the substance in the drawing that's changing. The enzyme is not changing, and the site of where their binding is occurring is the active site. How does this all happen is it's been referred to oftentimes as something called the lock and key, but today we really call it the induced fit. So if you look at this diagram over here and watch what happens with the two different little pieces here, the substrate's going to come along and bind to the enzyme. The enzyme is going to act on it or change its shape a little bit so that it can cause the reaction to occur, where I'm taking one product or one substrate and dividing it up into two products. The specific substrate has to bind to a particular active site on a particular enzymes. It's the only one that can fit. Now, it isn't like they're going out and seeking each other. It's that these molecules would be bouncing around inside the cell, and they would hit and they would fit into the little piece, and the reaction would occur. If the piece doesn't fit into, if the substrate doesn't fit into the enzyme, then the reaction can't occur, and it would just bounce back off, and nothing would happen. So enzymes are just random acts of running around, bumping into each other, and colliding. And then if they fit into the particular one, they're going to cause a reaction to occur. So basically, what is the gist of what enzymes do? Is they're basically going to bring an enzyme or substrate together, and cause a molecule to make a larger molecule as an endergonic reaction, or they can put stress on the substrate bonds and break them, causing an exergonic reaction where energy is released. So it depends on it's an endergonic or exergonic what the enzyme is going to be doing. So what are some of the properties? We've already mentioned that all enzymes are reaction specific. In other words, one enzyme works with one particular substrate. And so if we look at it this way, if I want to have a chemical reaction that eventually I produce product G, and it's going to take multiple steps to get there because there's never just one step of something in your body. It always takes multiple steps. I'm going to start with substance A and convert it into B and using enzyme 1. Then I want to convert B to C. Well, then I have to use another enzyme because otherwise it wouldn't fit. I get to C. If I want to get to D, I have to use another enzyme. D to E, another enzyme. E to F, another enzyme. And then F to G, another enzyme. So each reaction that occurs is specific to a particular enzyme that's going to work on it. If we look again at this diagram, again, here's a enzyme and a bunch of substrates bouncing around inside a cell. Not all of them are going to fit in. So only one particular substrate will fit into this enzyme where it will then reform its shape and then cause the reaction to occur. Again, we'll just watch it in a minute. This one's not going to fit. It's just bouncing, randomly moving around inside a cell for this thing to occur. It's going to continue to try, but it doesn't. It's not like it has a mind of its own or is actively seeking. They're just randomly motion molecules moving around. And if it hits one and fits, a reaction can occur. If it doesn't fit, a reaction's not going to occur. It's just going to keep continually bouncing. It's like a bunch of ping pong balls bouncing around each other until one fits into a slot, like a pinball machine, sort of. Another property of enzymes is that when they do cause a reaction to occur, they do not get destroyed, and that enzymes are very unique. So enzymes are unaffected by the reaction. They can be reused over and over and over again. So therefore, if you increase the amount of enzymes that are in a particular concentration, you're going to increase the amount of reactions that can occur. So the more enzymes, the more changes that can occur. You increase the substrate, the same thing can happen. You have more substrates that can get the reaction with that particular enzymes. Another property of enzymes is that they have an optimal temperature in which they work and an optimal pH in which they work. If it's too cold, the molecules slow down and they can't fit into the enzyme. It takes too much time. If they're too hot, it's going to change the shape of the enzyme. And then it won't be able to bind with its particular substrate. When we have an enzyme changing its shape, we call it being denatured, so it no longer fits with its particular substrate. And temperature can do that, and pH can also do that. If you have too many or too little H pluses, then the acids are going to denature that enzyme, and then you no longer get your nice induced fit. Now, normally all this is going to be occurring in humans at around 37 degrees Celsius. If you get too high or too low of that, then the proteins become denatured and they can't work. The same thing with pH. The normal pH for most of the reactions to occur is anywhere between 6 and 8 or in the neutral range. However, there are a few that like to work in a very acidic pH. But it is important that enzymes work in an optimal temperature and pH range. Enzymes also can be slowed down by a particular inhibitor. And there's basically two inhibitors we're going to talk about. We're going to talk about the competitive inhibitor and a non-competitive inhibitor. And so if you look at this first diagram here, we have an enzyme that's going to bind with this particular substrate on its active site. Well, competitive inhibitor is going to actually come in and block the substrate from the active site so that it can no longer bind and the induced fix can cause a reaction to occur. So it competitively competes for the active site, not allowing the substrate and the enzyme to bind together for the reaction to occur. A non-competitive inhibitor is one that doesn't act on the active site. It just attaches itself to another location on the enzyme, causing a shape change, and therefore not letting the reaction to occur. One happens on the active site, that would be your competitive one because they're competing for the active site. The other one is non-competitive because it's not on the active site, it's on another location, but it does pinch in and cause a change in the shape of the enzyme, therefore not allowing the substrate to bind to the particular enzyme. And so How do all these reactions occur? Do they just keep going all the time? That enzymes are just making, and breaking, or is there some kind of control mechanisms that says, all right, I have a certain level of enzymes, that's all I need. I don't need any more of it anymore at any given point, so I'm going to stop the production, or I need a large amount of it. And so we have the body has this very unique, and all living things have this unique feedback mechanism, and we call it feedback inhibition. And so what happens is if your body registers that you have enough, for example, of the enzyme G or the substrate G or the product G, then it will send an inhibitor all the way down to the beginning of where A is made of your first step, so that if A can't be made to B, then you're never going to get to C, then D, they need to make any more G's. And so in other words, it inhibits the production of this first enzyme, so therefore you can't go from one molecule to the next. And that inhibits the whole general process. Once, if you needed more of it, the inhibitor would be stopped and it would go back into production to produce the G. So this feedback mechanism can be turned on and off by inhibiting or not inhibiting the production of particular enzymes. How do we name enzymes? Enzymes usually end in ASE, and so whenever you see a big long word and it ends in ASE, you're going to recognize it as an enzyme, and therefore you'll know that it's going to speed up chemical reactions and lower the activation energy. You're going to know then that it works in a particular temperature and a particular pH, and you know that it works on a particular substrate. So for example, lactase is always going to work on lactose. Amylase is always going to work with amylase. Amylase is going to work on amylose, and sucrase is going to work on sucrose. So that does it for enzymes. Let's look at what we should have learned from this video. You should be able to tell me the function of enzymes in that they speed up chemical reactions and make things easier for reactions to occur. You should be able to look at a graph and tell me whether it's a reaction happening with enzymes or without energy. You can tell by the amount of energy that's being used. And in that case, then you can describe what activation energy is and how it affects enzymes. Can you, if I give you a picture of an enzyme, locate the active site, the substrate, and the products, and know which is the enzyme as well? Can you explain how the enzyme binds to a particular substrate, causing that chemical reaction, doing the induced fit? And can you explain the role of pH and temperature and the other properties on how they affect the ability of an enzyme to work? Can you tell me the role that inhibitors play in enzymes' functions, both of them, the competitive and non-competitive? And can